So thank you. Thanks to the organizers for inviting me here. Singapore is one of my favorite places in the world, so I'm really happy to be here. So yeah, I want to talk about one publication that was hopefully going to be published soon about metabolic drivers of species aging. And I'll point out it's a collaboration with Kostas Lysiotis at the University of Michigan and Brian Kennedy, who is obviously here. So this is my lab. It's been mainly led by Steve Pilley, Edgar Esparza, and Xuanyi Shi. And we're really interested in how age and sex affects metabolism in health and disease. So healthy aging, but also diseases of aging. And the great thing about being in the last session is I don't have to introduce aging, I don't have to introduce metabolism, because basically, I think Vadim had in his talk, lots of the promising potential gerotherapeutics target metabolism, whether it's caloric restriction, rapamycin, metformin. So I don't need to convince you that metabolism is important in aging. But despite that, it's not known how metabolism changes throughout life. So we decided to tackle that on, take that on. So what we do is we take a comparative biology approach to identify these metabolic drivers of aging. We look at lots of different species. Here's just some of them on the left. We do lots of different organs because we want to find out actually organ-specific changes. We do metabolomics, lipidomics, untargeted, targeted, and we hope to find some aging mechanisms for potential gerotherapeutics. So what I'm going to talk to you about today is just the first one. That's on bioRxiv now. You can look down there. It's undergoing revisions. And it's in black 6 mice. And we basically modeled this on the tabula muriscinus. So we had the same time points, the same diet, the same mice, same organs. So we could also use their data as well to combine it with our metabolism data. So this is a targeted metabolomics, is all I'm going to show you here. So these are the questions we're hoping to answer. How did it change through life? Can we identify gerotherapeutics? So okay, how does organ metabolism change throughout life in these mice? So this is all the data. So this is a Spearman correlation showing how each sample correlates with every other sample. So the more purple it is, the more it correlates with the other samples. And what's really nice is that you can see each organ, each sample in each organ correlates best with its own organ. So you get this really nice purple line down here, which gave us confidence in the data. That's nice. You'd expect organs to have metabolic differences. But despite that, you start to see some interesting trends. So for example, our two skeletal muscles here, the tongue and the quad, they correlate obviously best with themselves, but they also have some correlation between the two organs. And then our other muscle, the cardiac muscle, is also slightly purple here. You also get some correlation between this muscle. And then if you look at our two immune organs, we have the spleen and the thymus. You can see also they correlate best with themselves, but they also share some metabolic similarities between the two organs. So that's all nice. So now we can take all that data. This is now showing age here. It's on a U-map, so distance doesn't really mean anything. But you can see that the organs cluster quite nicely. We've got their ages in different colors in months. And you can start to see some interesting patterns. But the way we like to show it, because we want to, we obviously want to do this because we're interested in metabolism, but we want it to be useful to the community. So that even if you don't know metabolism, if you have a favorite organ or a favorite species or a favorite disease, you can go into the data in a user-friendly way. So we create nice websites that you can go in and really get out the data in a way that will be useful to you. So one way we do that is by showing these chord diagrams. So here on the left, we've got a log2 fold change from the earliest stage, and then we've got each line here is a metabolite. So if it's gray, it's not significant. If it's purple, at some point during life, there was a significant change in that metabolite. So then you can start to see, okay, maybe at least the brain in our hands didn't show that many changes, whereas the thymus has some great changes. So again, if we take our two skeletal muscles that I showed on the correlation, you can see the quad has a lot of changes in metabolites throughout life, whereas the tongue doesn't. And actually that's quite cool if you think about it, because one of the biggest problems with aging is that you get lower body muscle wasting. Whereas, again, it might sound silly, but no one talks about your tongue shrinking when you get older. So maybe there's some sort of metabolic drivers that protects the tongue from here, whereas the skeletal muscle here, the lower body one, wastes. So we're really interested in finding out, is there something in the tongue that's protecting it from wastage, or is it just use that, you use your tongue, you talk, you still eat when you get older, whereas you become more sedentary and sit down. So you also can look at the immune organs I spoke about, the spleen and the thymus. They also show different patterns. Again, this is the log2 fold change. So you can see there's a huge range in levels of metabolites in the thymus, which you don't see in any of the other organs. So then you can start to, okay, let's go and look at these two as a proof of principle. What's going on in the spleen and the thymus that's different? So if you look at those metabolites that are changed in the thymus that really crash, basically every single one of them is a nucleotide. So this is just 4 representative ones here. We've got the thymus at the top, the spleen at the bottom, just 4 representative nucleotides. And you can see that with age, you get this really big nucleotide crash. In the thymus, you don't really get that in the spleen. So then we worked with Sophia Liu at Harvard. She did some spatial RNA-seq on the thymus, and she showed that really there's proliferation markers of lots of different immune cells really decrease in the thymus as you age. So maybe that makes sense. You know, proliferation goes down, nucleotides going down. We don't know if it's cause or effect. It's probably changed in cell populations, but this makes sense that nucleotide levels are crashing as the immune cells are also crashing. And it makes sense biology-wise. You look at the thymus with age. Thymic involution happens in most species that have a thymus. So this in mice, you see with increasing age, you get a loss of structure. The thymus really just becomes, just loses all structure. You get fat infiltration. You also get a loss of cellularity. Also happens in humans. I think this is a beautiful image. This is not from us, this is from a paper a few years ago. You see that in the old human, you really get a loss of structure. You get infiltration of fat, buildup of fat, sorry, and the loss of the cortex and medulla. You also get a loss of cellularity. So these nucleotides crashing, I think, really give confidence in the data set that, okay, this makes sense from a known biology point of view, so then you can go in and look for things that are unknown. Let me just check how long I've got. Okay. So you can use this data set to go into your favorite organ and look for known things. Okay, you can also look at sex. So females in yellow and males in gray. This is the thymus and spleen. This is just showing the same data, just differently with the nucleotides. And you can see that females maintain their nucleotides longer than the males. They also crash to their levels a bit later. And this, again, makes sense from biology. Females undergo thymic involution later on in life. The whole, women have a stronger immune system as you age. So we think this is really interesting. This nucleotide crash could maybe explain this. Okay, so in the last 3 minutes, can we use this data to identify gerotherapeutics? And this is where Brian's lab came in. I won't explain aging clocks there, but we want to basically say, can we use metabolic clocks to identify drivers of aging? So not just predict age, but identify potentially important drivers of aging. So we worked with Max and Jakeem, they're both in the audience, and Brian to build a metabolic clock. I won't go into the details just in the interest of time. But basically, this is just in the plasma. You can predict age. This is the training set, the test set, just based on metabolite levels. And we did this for every single organ that we have as well. But the really cool thing is then, the way they built the clock, they did it on a minimum number of metabolites, so not on all metabolites. What's the fewest metabolites you need to predict age? So I'm just showing you the plasma here. You can see there's 8, all you need is 8 metabolites to predict age. And so maybe these are potential important drivers of aging in this organ. So I'm just going to highlight one of these, alpha-ketoglutarate, because we're in Singapore and Brian loves alpha-ketoglutarate. So you can then check in the plasma. It drops. That makes sense. This is great. This has not been shown before in metabolic data sets. This is just showing in the plasma and the bladder. So then we can actually look at the plasma and the bladder and say, okay, it drops, it really crashes alpha-ketoglutarate in the bladder. So let's look in the bladder system. This is just 2 bladder cancer cell lines. When you add alpha-ketoglutarate to the cells, they really drop the growth of the cells in the bladder cancer. So the clock has allowed us to look in the bladder to basically find that. But what about other metabolites, things that are less known? So we had a really promising hit that I actually can't tell you the name of it right now, just for potential IP purposes, but we thought this is going to be great. We're going to add this metabolite in whatever system, and it's going to increase lifespan, protect whatever. So we worked with Mary Simons at the University of Sheffield, intracerebral, just because it's quicker than mice, to do this. And you can probably tell from the way I'm introducing it, it's not going to show what we thought. But basically, so here's a curve. The dashed line is dietary restriction. So if you look at the black to the black, dietary restriction, the Drosophila live longer. That's great. We add our metabolite, and we basically kill the flies. It's a complete crash. So if you look at the dashed line here to the dashed line here, our metabolite basically takes it even lower than the regular food that the Drosophila are on. Here's the significance here. So we expect it to go the other way, but that's fine. It goes to the left. We kill things. We found something that kills flies. But at least we have a pathway now we can target a different way. So actually, it's quite exciting. Just to really show that, we did this in a couple of liver cancer cell lines. This is the metabolite in yellow. When you add the metabolite to the cancer cell line, it actually increases proliferation of the cells. So again, it's probably not a good thing during aging, even though it did crash the levels. So this is just one example. We're very excited about this. We've actually figured out how to potentially rescue this effect as well, mechanistically, but I don't have time to go into that. So here's just a summary. Metabolic aging varies between organs. That's important. And you can use metabolites via a clock to identify potential gerotherapeutics. Here's the QR code if you want to check it out. And this is everyone who helped. In bold is some of the data I showed. Obviously, big thanks to Brian, Jakeem, Max, Mireille, and Costas, and then everyone in my lab, and Sophia Liu, and here's the funding. Reach out to me if anyone wants to chat about metabolism, and I'd love to take any questions. Thank you. If no one has a question, I can tell you why my elbow's really swollen as well. In the poster session, I got bitten, and now it's swollen up, so don't go in the poster room. Yeah. I don't know, it might have been a student, I don't know.
They're very dangerous, the biting students. So the alpha-ketoglutarate decline with age.
Yeah.
Can you speculate on what's driving that?
Oh, maybe that's a question more for Brian. Alpha-ketoglutarate, it's not in every organ that we see the decline. What I showed you was the plasma and the bladder, so it's probably different in each organ. It maybe is in certain organs, maybe the TCA cycle is reduced, the activity of the TCA cycle, maybe glutamine metabolism down, is probably organ dependent. Thank you very much. Oh, there's another question.
Hi. I have a question about alpha-ketoglutarate as well.
Yeah.
Is there any organ that the alpha-ketoglutarate doesn't decrease with age?
Definitely.
Which one is it?
Oh, I can check after and I can tell you, yeah.
Sure.
It declines in, I think, maybe half of the organs. I just don't want to say the wrong one, but I can, yeah.
Okay.
Thank you.